And I will say we are going to be working with some equations. So if you have a calculator with you today or a phone with a calculator capability, that's awesome. You will probably need it. All right, we're not doing anything too advanced. Just your phone will be able to work too. But any questions going forth towards the end of the semester here? All right, well, I'm going to show you what homework you need. Yeah. I believe it's Saturday, the 18th.
Okay.
I have to double check, but I believe I wrote it down on the schedule.
In person?
In person,
Okay.
yes, unless something drastic happens with COVID.
Okay.
Any other questions? All right, I did put the study guide up for the final. All right, that's been up for a while now. The final is going to be mostly this new material. I think it's like a 60-40 split. All right, so the study guide I put up is for the new material. I did put it up ahead of time, so I will try my best to go back and take out any irrelevant questions as we go through the semester. As for the cumulative stuff, go ahead and look back at your old study guides. That's the best way to study for the cumulative portion of that. For homework going into Wednesday, I am going to ask you to watch this video on, I believe it's on sex selection when we're talking about evolution, because we're not going to have quite enough time to talk about this in lecture. And then there is a quiz this Friday, all right, and there's another assignment, all right, your last assignment. So this assignment is going to be due next Monday. After lecture today, you should be able to completely finish it and turn it in. I am going to ask that you print this assignment out and turn it into class on Monday in person. All right, because it's going to be a lot of equations. I want to see your work, so I want to be able to follow how you got the answers that you did. I put the assignment worth 10 points. All right, now we only have 9 points left that you can earn in the rest of the semester. So if you get all 10 points, I will consider that one extra 10 points an extra point on the exam. All right, so there's a little extra credit opportunity for the exam coming up there. All right. So we're getting close to the end of the semester. Only 2 quizzes left. Remember, I dropped your 2 lowest grades for those quizzes. Final assignment and then the final exam. All right, that's all that's left. So we're getting really, really close here. All right, so let's switch our thinking to population ecology. So population ecology, we kind of already went over what is a population. I wanted to define it even a little bit more specifically again. That way we remember what a population is. So a population, that's going to be a group of sexually reproducing organisms. All right, that's going to mean that they can interbreed, which means they are a single species. All right, so maybe it's a population of antelopes or a population of mice. All right, they are a single species. And a population is going to have an easy traveling distance between them. All right, so they're going to live in the same general area. Now, depending on what type of animal we're talking about, this distance can differ. All right, so the distance that mice can travel versus the distance that humans can travel, or the distance of caribou can travel. All right, they're vastly different. All right, so when we talk about a population of mice, maybe we're talking about a specific farmland. All right, if we're talking about caribou, maybe we're talking about this whole geographic region in Canada. All right, so it can differ depending on what species we're talking about. I threw this picture up here of the wolf. All right, there's 3 kind of populations, broad populations that we recognize in the United States for wolves. All right, but each population is going to have a gene pool. All right, and this is where we start to get into population genetics. So the gene pool is going to be all the alleles for every gene in a given population. All right, so if we just look at the gene for beetle color, all right, so that's the example that I put up here is beetle color. And if I want to look at the gene pool for beetle color, I can take a look at all the individuals in my population and see that I have these 3 distinct colors, all right, and they have different genotypes associated with them. All right, so this red one is big A, big A. This light yellow one is little a, and then this golden one is big A, little a. All right, so just as kind of a review, if I have 3 different colors but only 2 alleles, what type of genetics is represented here? What would I call this?
Like incomplete dominance.
Incomplete dominance, yes. So we don't have one that's totally dominant over the other. All right, if we only have 50% of one allele, it's going to have this intermediate coloring to it. All right, incomplete dominance. All right, so if I'm looking at the population genetics, I'm going to be looking at the genetic variation within a gene pool to see how this variation is going to change from generation to generation. All right, now typically speaking, the emphasis on population genetics is within one generation. We often say that this is the population genetics for this specific generation in this point in time. All right, but know that we can take population genetics and look at this generation after generation. All right, so just a brief review on genes, putting some new terminology on concepts that you should already know. All right, so polymorphism. Polymorphism is the presence of two or more variants or traits for a given character in the population. All right, we have seen polymorphism before, especially when we have a dominant allele and a recessive allele. All right, maybe it's eye color. Maybe one eye color is brown, one is green. All right, we have different variations for a singular gene. So a good example is this flower population down here. All right, it's the same species, but they show polymorphism. All right, because it has a trait for red flowers and a trait for yellow flowers. All right, so depending on which one is dominant here, let's say red is dominant, that might be big A, and yellow, it might be recessive, we'd call that little a. All right, so we've seen this before. We're just adding a term to this concept that we already know. All right, so we call this a polymorphic gene, or it has two or more alleles. Typically, we only see it as two alleles when we write out something as big A, little a, big A, big A. All right, that's only two alleles. All right, but that would be a polymorphic gene. Now, sometimes we get a monomorphic gene. All right, so this is when we have a population and this gene is only has one single allele over 99% of the time. All right, it can get this way through different evolutionary means. It could also occur this way through mutations. All right, a very common mutation is the single nucleotide polymorphisms, often called SNPs. All right, we know it as a point mutation. All right, different wording, same concept. This is a point mutation, or we also say it's a SNP. All right, it's the smallest type of genetic change in a gene, and it's the most common type of variation that we're going to see in human DNA sequences. All right, most of the variation we see among different people are going to be these point mutations, these SNPs. All right, to give you kind of an idea of how many of these point mutations might be in our genome, about 2,000 to 3,000 base pairs, all right, in length. An average of about 10 of those are going to be point mutations, are going to be these SNPs. All right, so because of these point mutations, these large, healthy populations has a high level of genetic diversity. All right, so mutations can add in genetic diversity. I put this example up here that we've already seen before, where we have a point mutation that leads to sickle cell disease. All right, so that's broadly some background. Let's talk about how would we analyze the variation in our population. All right, so if we want to look at a population and say, how many of these individuals have the dominant allele? How many have the recessive allele? What is their genotype? All right, we're going to look at frequencies. All right, and this is where we want to make sure we have allele frequency and genotype frequency, those definitions thoroughly understood. So if I'm talking about an allele frequency, I'm talking about one variation of that gene. All right, so maybe that's the dominant variation, or big A. Maybe it's the recessive variation, or little a. All right, so I'm talking about specifically one of those alleles. Now, if I'm looking at the genotype frequency, I am looking at how many, or both of their alleles put together. All right, so big A, little a, big A, little a, little a. All right, are they heterozygous? Are they homozygous? All right, so now I'm talking about genotype frequency. So remember, we have two alleles making up our genotype. All right, this is where we start to get into the math portion of our lecture today. All right, so I know a lot of people go into biology thinking, oh, I don't have to do math in biology. Biology has a ton of math, all right? Thankfully, in this course, we're sticking with very simple mathematical equations here. And we're going to look at the allele frequency and the genotype frequency. So if I'm looking at the allele frequency, all right, I'm looking at the number of copies of a specific allele in a population. All right, so maybe I want to know how many copies of big A are in a population, or how many copies of little a are in a population. All right, I'm looking at one singular allele. All right, and I'm going to divide that by the total number of all the alleles for a gene in that population. All right, different from genotype frequency. All right, remember now we're talking about both alleles put together. All right, the number of individuals with a particular genotype in a population, all right, is going to be divided by the total number of individuals. All right, so if I want to look at big A, let's see, I have 2 individuals in this population that show big A. I would divide that by the total number of individuals, and it looks like this individual is missing one allele. All right, we'll assume this is little a. All right, but we have 1, 2, 3, 4, 5, 6, 7, 8, 9 individuals. So I would take 2 divided by 9. All right, now this is a very simple way of calculating allele and genotype frequencies. All right, we're going to see when we look at these 4 o'clock plants, all right, this is the example that your book uses, that things can get a little bit more complex. All right, so if we look at this example, we're going to calculate the allele frequency and the genotype frequency, all right, of 100 of these plants. Now we are given that 49 of them are red flowered with the genotype big R. All right, 42 of them are pink flowered with the genotype big R little w. And in fact, normally we'd say big R little r, but they're using W for the recessive allele in this case, so it'd be big R and W. All right, and then 9 white flowered plants, which are WW. All right, they're giving us how many individuals have each genotype in our population. So they're making it fairly easy for us. All right, so remember when we're talking about allele frequency for a diploid species, these individuals have 2 copies of an allele. They're homozygous, it's 2 copies of the same allele. Heterozygous, it's 2 copies of a different allele for each. All right, so let's look at the frequency of a specific allele. And in this case, we're going to look at this allele for the white color. All right, so if we look at the allele for the white color, and we know the genotypes of our species, we can plug it into our equation here. All right, so R and W, all right, that genotype we know has 1 instance of W, all right, and we know that the other genotype that has W is this homozygous genotype for WW. All right, now we know that there's 2 instances of the W in this genotype. All right, that's why we're multiplying this second one by 2. All right, now we're going to divide it by the total number of alleles in our population. So each individual has 2 alleles. So for each genotype, we take the number of individuals times 2 to get the total number of alleles. All right, so we're going to plug that all into our formula. We have 42 individuals that are heterozygous. All right, we have 9 individuals that are homozygous for this white coloring. Right? We're going to times that by 2 because we know they have 2 alleles for the white coloring. Right? Divided by the entire population of alleles, and eventually we're going to get 0.3 or 30%. Right? So 30% of the population of these 4 o'clock plants have the allele for white flowers. All right, now we can take it a step further and look at the genotype frequency. All right, so genotype frequency, we're looking at 2 alleles together now. All right, and this is a little simpler in that we know how many individuals are homozygous for this white coloring, which is 9. We divide it by the entire population of individuals, which is 100, and we get 9%. All right, 9% of these individuals have the genotype for homozygous white coloring. All right, so that's a lot of math, and we can kind of think about how we're going to calculate genotype and allele frequencies by using the Hardy-Weinberg equation. All right, so typically what we just did there, is a little bit more complex than it needed to be, and we can simplify that using the Hardy-Weinberg equation. All right, so this equation is going to describe the relationship between allele and genotype frequencies when a population is not evolving. All right, so we are at equilibrium. There is no evolution happening in our population. All right, so if we have a gene that exists in 2 alleles, we'd say P plus Q equals 1. All right, so P is representing one allele, Q is representing the other allele. All right, now again, this is a population that's not evolving. All right, so this population is going to remain the same generation after generation after generation. No evolution is occurring. All right, so if we look at our 4 o'clock plants here, all right, and we just learned that the Hardy-Weinberg equation is P plus Q equals 1. This is telling us the allele frequency. All right, allele 1 and allele 2, combined together, they have to equal 1. Now we can modify this equation to tell us the genotype frequencies, all right, where we get P squared plus 2PQ plus Q squared equals 1. All right, so that's the genotype frequency. It's P squared, right, because we have 2 RR homozygous individuals for the red allele in the RR homozytes, right? We have 2PQ because we have 2 of each allele, all right? We're going to times that by 2. And then we have Q squared because we have 2 white alleles for the white homozygous individuals. All right, so using this, we can calculate out that R is 70%, white is 30%. All right, we can then plug that into our equation here once we know P and Q, plug in our numbers, and get the frequencies for all of those genotypes. All right, so I just wanted to step back and make sure we understand where we are getting these numbers from. All right, so remember, to create an individual, we need genes from our mom and genes from our dad. All right, egg and sperm, they're coming together to create a zygote. All right, the egg is going to carry one allele, the sperm is going to carry another allele. So if we go through, we can essentially make a Punnett square, where if we know the sperm is going to be 60% big A's, 40% little a's, all right, 60% big A, 40% little a for the eggs as well. Go through, multiply them out, all right, and get the probability of each instance of making a zygote in our population. All right, now the biggest takeaway here is that these genotype frequencies have to equal 1. All right, so notice for the heterozygotes where it's big A, little a, all right, we added them together to get 0.48. All right, and this is just another way to talk more generally about how we're coming up with this equation here. We can look at it again, look at egg and sperm, look at the probability of each zygote occurring. Now I'm not going to go through this step by step, but I put this in here for your reference if you needed a little bit more of a visual there. All right, so that's the background for the Hardy-Weinberg equilibrium. All right, it's kind of a dry subject, but it can tell us some very interesting things about our population. However, there have to be different conditions that we have to meet in order for a population to be in equilibrium. All right, remember I said there is no evolution happening. All right, that must mean that there is no new mutations occurring. Natural selection is not happening. The population is so large that our allele frequencies do not change due to some random sampling error. No migration is happening between different populations, right? And the mating is totally random, right? We're not mating based on how an individual looks, for example. It's totally random, right? Now, this very rarely occurs in nature, right? We hardly ever see all these conditions being met, right? But we still find Hardy-Weinberg equilibrium useful because it gives us a baseline, right? If we know what happens in a population if we assume no evolution is occurring, then if we go back and calculate the same things over and find that the frequencies are not in equilibrium, right, we know that there's some sort of evolutionary mechanism at work. And we'll go over those evolutionary mechanisms on Wednesday. Right? But big takeaway, no evolution in our population when we're talking about Hardy-Weinberg equilibrium. All right, the frequencies of our alleles are going to stay constant through generation after generation after generation. All right, so the probability of me drawing a big A from our population and the probability of drawing a little a from the population is always going to be big A, 60%, little a, 40%. It's always going to be the same. All right, population is an equilibrium. I put several slides of this on there because I really wanted to drive this point home. All right, so how do we analyze genetic variation in a population? We're going to look at frequencies. All right, allele frequency, genotype frequency, and also use our Hardy-Weinberg equilibrium. All right, let's do an example problem together. All right, so this is going to make way more sense, and I hope it's not going to be quite as dry. We have a population of deer here, all right, in which we have a population of 200 deer in which 65 of them are white. Now we know white is the recessive allele. All right, now I want to know what is the allele frequencies and what are the genotype frequencies. All right, so given this information, and this is something that I could ask you on an exam to try to calculate out, right, I want to know the allele and the genotype frequencies. All right, so let's start with what we know. In a population of 200 deer, 65 of them are white. All right, so if we know white is the recessive allele, what is the genotype for our white deer? Little a, little a? Yes, little a, little a. All right. All right, we know it's little a, little a. We know this, all right? It has to be little a because it's recessive and the phenotype is showing through. We know it's homozygous recessive. All right, now what might be the genotype for the brown deer? Yeah, big A, or big A, little a. We don't exactly know, but we know it's one of the two, right? Because brown is dominant, it has to be one of those. All right, so we know the white deer is little a, little a. All right, we also know our Hardy-Weinberg equilibrium equations. All right, and you should have these memorized by heart going into the exam. All right, we know P plus Q equals 1 is the allele frequency. P squared plus 2PQ plus Q squared equals 1 is the genotype frequency. All right, we know this. Now, if we know that 65 out of 200 of the deer are white, all right, we can divide our population, and we know that the frequency of this particular genotype, little a, is 0.33, or 33%. All right, so just from the information that we were given, we can easily determine the percentage of this genotype in our population. All right, 33% of the deer are white. All right, so now we have the genotype frequency, but how exactly do we get the allele frequency? All right, well, let's plug it into our equation here. All right, so our genotype frequency we just calculated for little a, we know is 0.33. All right, so if we plug it into our equation, essentially we get 0.33 squared. All right, so if we were to take the square root of that, all right, we would find the instance of just one singular A. All right, so again, we have this 0.33 is the genotype frequency. I'm trying to find just the allele frequency of 1A. So 0.33 is 2A. I want to know the instance of 1A. All right, I'm going to take the square root of that, and I'm going to find that little a in our population is present 57% of the time, right? 0.57. All right, this is probably the most complex math you'll do in this whole series of equations. All right, so I know that A equals 0.57, all right? How might I find the instance of capital A? Subtract it from 1, yes. All right, we know P plus Q equals 1. Another way of saying that is big A plus little a equals 1. All right, we'll just subtract from 1, and we'll find P, all right, or big A. And we know that that equals 0.43, 43% of the alleles in our population are going to be the dominant allele, big A. All right, so we just found our frequencies for both of our alleles, big A and little a. Now it's just a matter of plugging in the numbers. All right, so we know the allele frequencies. We can plug it into our genotype frequency equation. All right, we can plug it in. We can calculate out the math here. All right, and we can find exactly what our genotype frequencies are. All right, for big A, big A, big A, little a, little a, little a. All right, so it seems like a lot of work. It seems like it's really complex, but it's really not. All right, you just gotta step back and think about what do we know, and how can I use that information with the equations of the Hardy-Weinberg. All right, so in a population of 200 deer, I only told you that 65 of them were white, which are the recessive allele. We were able to figure out the allele frequencies and the genotype frequencies. All right, so I would like you to do a practice problem on your own and apply what you just learned. All right, and then we can come back together and talk about it. And if you have questions arise as you're calculating this out, let me know. Feel free to work together. Maybe somebody has a better calculator, I don't know. I'm still in a therapy coma.
Nice. I watched the Confederate. Wait,
We'll go over it together.
awesome. What part? Okay, so dead center and stay up like 2 hours. says never attempt to apprehend a fugitive yourself.
Oh, but
In my hometown. He was sentenced to first-degree sexual assault of a child.
no.
Somehow ran out of prison. He's
It
living
sounds
in
like we're
Trenton.
talking about allele frequencies over here.
This is kind of a little creepy.
I got some kind of weird frequencies.
I mean,
We'll go over it together as a class. Don't
I
worry.
actually understand.
what? I asked you guys if you understood.
All right, so we know what the genotype and allele frequencies are then,
So
right?
what is
Oh my gosh, she's on her way back. She's on her way back right now.
that sound correct?
I don't remember the numbers off the top of my head. Are we still calculating out our all data over here? Alright.
Is that a good thing for Ed?
Well, I literally just said it to make me.
All right, so it sounds like most of you have things calculated out. These are the answers. How did we come to this? All right, so we were given how many of each genotype exists, right? All right, so what's our total population? 500. All right, we have a total population of 500. All right, so in this case, I did not give you the total population, but you were able to calculate it out because you know how many individuals of each genotype there were. All right, so genotype frequencies in this case is relatively simple. Right? We want to take the number of individuals. Let's start with big W, big W. All right, that's 320. Divided by the total number of individuals in our population. All right, and what did we get? What's here on the screen? 0.64, yes. So to get that, we know 0.64 or 64% is our frequency for the genotype of big W, big W. All right, so we can go through and do this for the other ones as well. So let's say the heterozygous type here, we had 160 over 500. And what did that equal? 0.32, yes. Again, right up on the screen there for you. 0.32 or 32%. All right, for the heterozygous genotype and the homozygous dominant. We only had 20 individuals out of 500. And that equaled, what is that, 0.04. Or 4% for that genotype. Alright, so genotype, pretty easy to figure out in this case. Alright, now we want to figure out the allele frequencies. Alright, so in this case, they kind of went about it in a different way. There's multiple ways to come to this answer here. So they added in the number of, let's look at this. We know that big W is homozygous, right? Alright, so we have big W. We know 160 are going to be also big W in the heterozygous. We added that together, we divided it by 1,000. Alright, remember we have 500 individuals, right? But each of those individuals have 2 alleles, so we'd have 1,000 alleles in our population. Now another way that you could figure out these allele frequencies is by using the Hardy-Weinberg equilibrium. Right? And I ran out of room to write here. But remember, P squared equals 2PQ plus Q squared equals 1. Alright, so let's start with the big W allele. Alright, and we know that P squared, which would be homozygous dominant, right, is 64%. Or 0.64. Right? All we'd have to do is take the square root of 0.64 and we'd figure out the allele frequency for big W. Right? So that's another way that you could come to the same answer. Right? Now a good way to double check to make sure your math is correct is to make sure all of these add up to 1. Right? So allele frequencies will add up to 1, genotype frequencies will add up to 1. All right, any questions on how to calculate that out? Any sticking points, anything we don't understand? All right, so I have another practice problem for you. All right, and this is very similar to, again, something that you might see on the exam, and I might ask you to calculate out. So go ahead and give this a shot. If you need help, let me know. Anybody need more time on this? All right, let's solve this together. What do I know? What is the question telling me? What do I know out of this question?
40% are W, little W.
What was that?
40% are little W, little W.
Yeah, 40% are white. White is the recessive, all right? If they're expressing that genotype, we know they're homozygous recessive. So we can say little b equals 40% or 0.4. All right. Anything else that we can deduce out of this question just off the bat?
Well, you know the frequency of the dominant allele because P plus Q equals 1, so it would be 0.6.
Exactly. Yeah. So let's see here. Dominant allele? Is that what we're looking at?
Yes.
Let's see here. I have something different. How did we get that? How did we get to that?
I just used the allele frequency equation. So P plus Q equals 1.
P plus Q equals 1.
So we know the recessive frequency is 0.4.
Is it 0.4? We know the genotype for the recessive is 0.4. If we had to figure out the just for 1B, what would we have to do? We take the square root, yeah. Take the square root of 0.4, all right, and that would give us 0.63, and then we could plug it into this equation. Exactly right. Yeah. So if we say, let's see, little b is Q, right? So b, we could go 1 minus 0.63. All right, that would give us 0.37 for big B. All right, so right there, we know the allele frequencies. All right, pretty simple, pretty straightforward to get to that one. Now it's asking us the percentage of butterflies in the population that are heterozygous. How might we figure that out? What other equation do we know? Squares. Nobody? What is the other equation that we know?
E squared plus 2P plus Q squared.
Yes, exactly. B squared plus 2PQ plus Q squared equals 1. All right. Now we know little b, all right, is 0.63. We know big B is 0.37. All we have to do is plug that into our equation, do the math, and then we can get the frequencies. All right, so if I want to figure out the frequency of heterozygous butterflies, what is that frequency? What did we come up with? 46%. We could round it to 50%. However, you decided to round it there. But yeah, 2 times 0.37 multiplied by 0.63. All right. Get that, we can round it up to 0.46 or 0.5, depending on how you round it. That is the frequency of heterozygous butterflies. All right. Now we can use that same equation to figure out the frequency of homozygous dominant individuals. And how do we do that? Plug in the numbers. We're going to square it. Yes. So let's see here. If we're looking at homozygous dominant, right? The dominant allele is 0.37. You take 0.37 squared, which would give us 0.14. All right, not only on the exam, but the assignment 4 that is due on Monday. Right? Assignment 4 is all Hardy Winder. All right. Get out 2 minutes early. I did put this additional resource if you're a visual learner. I thought the Amoeba Sisters did a really great job of explaining this.